Hello my dear students, I hope all of you are doing very well. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. So, we are learning pharmaceutical biotechnology and day by day we are gaining knowledge, the updated knowledge, the latest information in pharmaceutical biotechnology, how the world is getting changed by advancement in this field. Today, I am going to tell you something very interesting thing, and after listening this lecture, your understanding in insulin, diabetes, diabetes type 1, type 2 and all the things associated with your sugar, everything will be very much clear, and you will have a very concrete, you have a very strong that is a base in particular this area. So, I am going to tell you about the insulin, but before that I will tell you the entire story, how insulin was actually developed and why it is important. So, what are the things we are going to learn today? I will tell you first a basic introduction to insulin, then I am going to tell you how you can produce insulin by using RDNA, that is recombinant DNA technology. Then I will tell you about few marketed formulation. So, I hope all of you might have seen these pictures or these things in your home or any of your relatives or people nearby you. So, this is basically, this one is basically a machine, biosensor is here. By using this biosensor based instrument machine, you can detect how much amount of glucose is present in your that is blood. Okay. And all of you might have also seen this kinds of pen, where insulin is actually given to the patients, those people having high sugar level, to reduce the sugar level, we are actually giving insulin. Now, all of you might have seen that it is actually given in subcutaneous way. Now, what is subcutaneous? As a student of pharmacy, you should be aware that intramuscular, subcutaneous, intravenous and intradermis, there are lots of differences. Subcutaneous means in the subcutaneous region our the insulin will be that is put it, and from there it will be get that is diffuse our entire bloodstream. Now, from where this insulin is coming? Insulin is actually coming from the pancreas. So, if you just zoom or if you just see your entire GIT, gastrointestinal tract, you will be able to see here in this area there is a very interesting organ called pancreas, and if you further zoom this pancreas, you will be able to see that in that pancreas there are various kinds of cell like alpha cell, like beta cell. Okay. So, the insulin, it is actually produced by the beta cells of pancreas here. Okay. So, if you see this this insulin, here I am trying to tell you the insulin. So, if you see, if you zoom on such insulin, if you zoom on such insulin, you will be able to see in 3D this kinds of picture. This is the actual 3D picture of insulin. Now, what insulin is doing? This is basically controlling the amount of glucose in your blood. You see, when you take some food, basically if you when you consume glucose, obviously glucose level will increase in your blood. If glucose level may increase, there will be some issue, the patient will face some problem. That is why insulin will reduce the amount of glucose, it will control. Okay. So, here you can see here from your this pancreas, insulin is actually getting produced, and side by side from your that is a stomach, glucose is actually coming. So, what insulin is doing? Basically insulin is actually coming when your this thing called pancreas release a huge amount of insulin. Insulin is actually coming to your cell surface. There are some insulin receptor. First it will bind with the insulin receptor, then it will give some signal. To another receptor, and that receptor will allow the glucose, whatever the glucose you are observing, this glucose, to allow this glucose to enter into your cell. In that cell, all of you are aware, there is a long process of metabolism. You have studied all the process of metabolism, how glucose is actually metabolized, and then glycogen and other things are actually get synthesized. Okay. Basically, insulin is actually helping this receptor, that is a GLUT4 to enter glucose into your cell. Now, in maximum cases, the whatever the things I have shown you here, in some scenario, such kind of good things are not happening. At the beginning of my lecture, I have told you, I have told you here that these kinds of instruments are much more popular. Why? Because those people having some problem with insulin production in this area, those people having some problem that is insulin production in this area, what will happen? The insulin production will be get reduced. There will be no insulin. If no insulin is actually coming here, or very less amount of insulin may come here, what will happen? There will be no any signal to the GLUT4, so that glucose level can come, glucose cannot enter. Okay. So, glucose level will increase. So, after that time, when you will test somebody's glucose level, you will be observing he has high level of glucose in his blood. What are the complications? The patient will suffer from white loss, then frequent urination, irritability, blurry vision and fruity breath. This kinds of problem will happen. These things are actually calling as a type 1 diabetes. Many of you might have seen or might have that is come across about the another term called type 2 diabetes. Sir, what is type 2 diabetes? In type 2 diabetes, the problem is actually happening here. Here your insulin, whatever the insulin it will produce, that insulin have to bind with this receptor. Insulin have to bind with this receptor. Now, the problem is happening, insulin cannot bind to this receptor. This receptor actually getting mutated. There are lots of reasons behind this mutation, or there are lots of reasons behind the resistance. Sometimes we call it as a resistant. Okay. So, insulin cannot enter. So, if insulin cannot bind or cannot enter in your this entire cell signaling area, so no signal will be happen. Due to no signal, this receptor GLUT4, it will not able to allow this glucose to enter into your cell. Ultimately, again glucose level will rise. What are the complications? What are the symptoms? Again weight loss will be there, excessive thirst will be there, then unceasing hunger will be happen, headache, dry mouth, these kinds of things will happen. So, that is why my dear students, you should be very, very careful. If you face some kind of problem, you should be immediately you should meet to your doctor. Now, here is the entire summary, whatever the things I am trying to tell you. In healthy patients, from your pancreas, insulin will be produced, it will bind to the that is a insulin receptor, and then glucose will be able to enter. In type 1, pancreas will be failed to produce insulin. Okay, there will be, you can see here insulin is present, here is no insulin is present. So, less amount of insulin will be produced, or your pancreas will be not able to produce sufficient amount of insulin. And in type 2, here resistance will happen with the receptor. This receptor will be not able to bind, or it will not able to allow the insulin to enter. Okay. So, ultimately glucose level will increase, here is also glucose level will increase, and here insulin level will also increase. This is a summary of our entire understanding of diabetes mellitus, type 1, type 2 and insulin. Now, this story is actually starting from 1800. Those days, when early time those days doctors got to know that yes, there are some issues, patients are actually suffering from such kind of issues. So, it was actually used to believe that the average life expectancy after the diagnosis of this disease, it will be only 1 year. It means, anybody got diagnosed with such kind of issues, such kind of problem, whatever I have shown you here, such kind of symptoms or such kind of problem. So, life expectancy will be 1 year. And then in 1840, a great scientist, he actually one doctor actually come out with a that is a few observation. He said that his patient was affected about 12 months ago with a pain in the body, followed by weakness, excessive urination, 6 times normal output, then sweet thirsting urine, great appetite and constant thirst. So, the symptoms are actually used to so much clear. with those time. And after that when they have diagnosed their patient, they have found the patient with diabetes having high sugar level, okay, high sugar level. And then those days there was a very famous that is a theory or famous that is a therapeutic option called starvation dieting. They have found that if somebody is taking high amount of glucose, that means food, if somebody is taking food, obviously he is taking glucose along with his food. So, if the person or if the patient may reduce their food diet, so what will happen? The glucose level will decrease, and if glucose level will decrease, then what are the complication is supposed to happen? It will not happen. So, you can see here is a picture, this person it was a before and this is the after. Okay. So, you can see due to this starvation dieting, due to taking the that is a less amount of food, people used to die due to starvation. Okay. So, shortly before the discovery of the insulin, prominent American physician Frederick Allen and Elliot Joslin, they advocated for severe fasting. Okay. And those days there was no insulin. So, people used to believe and people used to follow this methodology. But you can see this the situation of this person after doing starvation. After that lots of good things was happened. What was happened, sir, after that? In 1889 pancreatic diabetes was actually discovered. Then in 1921 somebody discovered penicillin. Insulin. Somebody discovered insulin. After that, they got to know the same year that how we can purify this insulin. And after a long time, in 1959, type 1 and type 2 diabetes actually got to clear to the people. Then glucagon was also introduced in 1961, and then after in 1966, first pancreas transplant was happened. Those days, scientists and people, they were so much convinced that yes, pancreas having some that is if somebody having some complication with pancreas, then insulin production will be low. So, if we transplant this pancreas, if we transplant this pancreas, then it will work, and it was successful those days. And after that, in 1967, laser treatment for that is diabetic blinding was happened. Then in 1976, A1C test was developed, and then the main good thing was happened in 1978. First human insulin was synthesized. This was a great discovery. I will tell you more about this. And then in 1976, insulin pen was developed, and then from 1990, insulin was taken through external pump, and in 2006, first inhaled insulin was actually approved by FDA. There are some issue with this molecule called insulin. It is a big molecule, it is a protein basically. You cannot take it orally, because it will have there will be lots of complication, or it will be not able to pass your intestinal barrier, okay, due to its big size. And then in 2014, stem cell or isolate implant was actually happened. This is a short story, and right now how we will produce, or what is the way of the production of the insulin. You have seen in my entire 13-minute lecture that how insulin is playing a big role. Diabetes type 1, diabetes type 2. If you want to stop diabetes, you have to take insulin, especially for type 1. Okay. Insulin is very important. So, ultimately from where we can get insulin? They have done a lots of research. They have found, the scientists have found that if you can take the pancreas from pig, okay, pig also having pancreas. If you take out the pancreas from pig, and if you remove the insulin, and if you purify that thing, and if you apply that thing to the patient, so what will happen? It will work. So, at the beginning days, at the earlier days, scientists used to so much dependent on pig. Okay, you can see here the picture in 1950, pig pancreas actually collected. Here you can see this is a pig pancreas, all the pig pancreas. From that pig's pancreas, 4,535 kg of pig pancreas they have collected. From that only 0.45 kg insulin was actually found. See, you can imagine how much amount of peaks were actually sacrificed to collect this thing. So, that is why it is a very big issue. Now, due to advancement of biotechnology or due to advancement of recombinant DNA technology, today we can now genetically engineered bacteria produce insulin. Right now, we are not actually sacrificing pigs, we are actually not sacrificing the pigs, we are actually using bacteria. We are actually using bacteria to make insulin. Now, the question is how? The answer is here. The answer is here. It is very simple process. We are actually using recombinant DNA technology. Whatever I am teaching you since my last few days, that RDNA genetic engineering recombinant DNA technology. By using this process, very easily you can produce insulin. Now, what is the steps? So, at first, here you have pancreas and you are aware the genomic information of pancreas. The genes which genes or which DNA containing the desired gene, it is extract for pancreatic cells, means we in our pancreas we have alpha cells, beta cells, islets of Langerhans, etc., etc. So, the area or the particular cells which are producing insulin, you have the genetic information. So, what you have done? This is the chromosome, this is the DNA strand, and from that you have tried to take out that particular segment of your DNA, which DNA containing the desired gene is extract from pancreatic cell. Basically, the insulin producing or insulin gene. Okay. In one side, you have done all those things in one side, and on the other side, you have taken one bacterial cell, and from in that bacterial cell, you have taken the you have worked with the DNA, that is plasmid vector, and by using the restriction ribonuclease enzyme, you have cutted the plasmid, and you have inserted your insulin gene in that plasmid, and then by using DNA ligase you have fixed it up, this one, and then this DNA it will call as a recombinant DNA, RDNA. This will call as a recombinant DNA. This recombinant DNA will be inserted back into the bacterial cell. Here you can see in this bacteria there are also bacteria. You are actually inserting those recombinant DNA to the bacterial cell. There are lots of process, electrophoresis, lots of processes are actually here, electroporation. Okay. You can follow any of the process, and then when this bacteria is here, they will translate, they will translate, translation will happen. When they will translate this information, it will produce insulin. Then very easily you may just purify the produced insulin from here, and then you may give this insulin to the patient. So, genetically engineered bacteria multiply and express the insulin gene to produce this thing called insulin. Whatever I have told you here, same thing I have written in a different format. So, any of this picture you can take for your examination. Okay. In step 1, we have bacterium, plasmid, and in that plasmid we are actually cutting this plasmid, we are actually inserting human insulin gene in step 3. In step 4, engineered plasmid actually insert into a bacteria, and that bacteria will produce huge amount of insulin. Okay. The process is very simple. Now, if I tell you step by step, first step is the preparing, where we are actually isolating the DNA from human cells. Okay. Human gene actually isolated, the mRNA actually taken out from the cells to isolate islets of Langerhans. The messenger RNA, that is mRNA, is a molecule of RNA that includes the chemical blueprint for the protein product. This isolated gene contain the code of human DNA for the production of the insulin, whatever I have told, and then this plasmid of the bacterial cell actually taken out from the cell. And note that E. coli bacteria actually widely used. E. coli is very easy, very good, and it is not so much harmful and very easy to use. That is why we are actually using E. coli. So, in the second step, what you will do? You will cut. How you will be cut? How you will cut these things? So, you see the plasmid of DNA of bacteria actually cut out, producing plasmid ring, which is actually empty segment of the DNA. Here is restriction enzyme or restriction endonuclease enzyme. This will actually cut the DNA in a specific recognition of nuclear sequences. Okay, it is called as a restriction site. The segment of DNA actually known as the sticky ends. These things will be this area will be called as a sticky ends. In third step, what we will do? In third step, it is very simple. Whatever the human insulin gene you have developed, you are going to insert that thing. So, with plasmid ring open, the gene obtained from human cell that contains the code of protein responsible for production of the insulin actually insert, and then human insulin gene actually combined with the bacterial DNA plasmid. Okay, very simple. And lastly, in the fourth step, you are going to insert this recombinant DNA. This when it will be completely that is attached now, like this recombinant plasmid, you are going to that is insert into E. coli. Okay, this is the E. coli. After entering, after that is joining or after inserting this plasmid, you can see in this empty place recombinant plasmid actually taken. And then what will happen? The cells need some nutrients in order to grow and divide. So, while they live, bacterial cells process turn on the gene for human insulin, and insulin actually produced in the cell. When bacterial cells reproduce by dividing, human insulin gene also reproduce in the newly created cell. Okay. So, here are few examples of marketed formulation like Humulin. You can see they have mentioned recombinant DNA origin. So, this Humulin whatever you can see in your screen, it was actually developed by using recombinant DNA technology, whatever I have shown you here in my entire lecture. There are other marketed formulations are also available, for example, like this Atropid. I hope all of you are aware about the brand name and generic name. So, you see every pharma company, this was actually developed by Irili. They have Humulin, and this pharma company, this is Novo Nordic, their brand name is actually Atropid HM. This is also that is insulin. Then another Novolin, Novartic, it is another formulation, and here you can see Sigma Aldrich. They are also they have also insulin. It was actually also recombinant insulin, and they actually expressed on yeast. We have in my lecture I have told you we are actually using bacteria E. coli, but here they are actually using yeast. Another example, this is insulin H bio R40 IU. It was actually developed by Cedico Pharmaceutical Company. I do not know from where it is. They are claiming this is a 21st century's that is a company. Another example I have here you can see this is the pen. So, this is called Capricorn Scientific Pharmaceutical Company. They have also insulin human recombinant, and they have also mentioned the storage. You can see at a very low temperature you have to store these things, because these are protein. At high temperature, all of you are aware, at high temperature proteins will get degrade, there will be denaturation and other problems will be happen. So, the protein will be not able to sustain in a very good condition. And if I tell some overview of all the that is insulins, all the market formulation I have told. So, you can see Sanofi, they have first got approval in 1997, their name was Insuman. Another was that is Novo Nordic, they got approval in 1991, and Irili got approval in 1982. You have seen what is the this is the Irilis insulin market formulation. And if I tell more, Sanofi actually producing by expressing on bacteria, and Irili also they are expressing bacteria, but Novo Nordic is actually expressing on yeast. So, trade name Insuman, this is Novolin, and this is Humulin. In exam you can write, and if I tell something more, formulation regular or basal or premixed in the both the way you can give. So, this is something about our today's learning. Today we have seen that how you can how insulin can play a very big role in our that is a human that is our body activity. Insulin is very important to control the that is a glucose level or sugar level in our blood. Without insulin, glucose level will increase, it will create a lots of problem in our body. So, and ultimately many of the cases you have seen people actually die due to get a high amount of glucose level. So, how can we control? By using insulin, and how you can produce insulin I also have told you by using the recombinant DNA technology, and at the end we have seen few of the marketed formulation. In next day, tomorrow I am going to tell you something much more interesting thing about PCR, polymerase chain reaction. Right now, I hope many of you might be listening this word called RT-PCR, because this is the COVID pandemic era when we are recording this lecture, and RT-PCR is so much popular. RT-PCR is highly reliable. So, for RT-PCR, it is basic actually starting from PCR, polymerase chain reaction. Okay. Sometimes for RT-PCR we call it as a real time polymerase chain reaction or reverse transcriptase polymerase chain reaction. So, how RT-PCR is involved in COVID, how polymerase chain reaction changed the entire field of our biotechnology, entire thing you are going to learn from me on our next lecture. I am going to tell you lots of important things, that what is PCR, principle, requirement, instrument, 3 aspects of PCR, things what you should that is things to try if your PCR does not work, example, advantages and application. So, next day we may learn these new things. So, thank you all of you, thank you for joining with me. I am very happy that you have listened the entire lecture. I hope you are learning a lot of knowledge, and in future you will be able to use those knowledge for your research and your studies. So, thank you all of you, God bless you all. We may meet in our next lecture. Thank you.